Alkan to this special seminar. It is essentially almost midsummer. So we have a truly distinguished speaker with us. And I must admit that it's by pure chance that he is with us today, because we have communicated by luck, and he mentioned that he would be here during this time of the day, of the year essentially, and we invited him to give us a talk on essentially what he has been doing for the past 30 years. Okay, and I will give a very brief introduction of Şefik Alkan. He's a very old friend of mine, actually, and his lovely wife, Wanda. They are all old friends from Hacettepe, essentially, but he left almost 30 years ago, and he went to Switzerland. He worked at the famous Basel Institute of Immunology for a long time, and in close collaboration with drug companies, essentially. Yes. Then afterwards he went to the States. I'm sure he can give a better description of his past, but I'll just pinpoint some of the moments in his CV. And he went to the University of California, San Francisco, for a number of years, and 3 years. And now he was associated with the Medical College of New Jersey.
I have a slide.
Okay, so I won't go into detail because I know it's very complicated. I've been following him.
So complicated.
Okay, so I'll leave it up to him. But he is going to talk on I'm sure, a very interesting topic on modern approaches for novel drug discovery. He's an immunologist and a druggist. What should I say?
Stop drug.
Yes, we're happy to have you.
Thank you. Thank you very much. Very kind invitation. I'm so happy to be here in the middle of summer. What are you doing? Really beautiful city. And we had a congress 2 days ago. Guess how many people attended? 8,000. Compared with this distinguished audience. Anyway, so what I want to do is give you a glimpse about the drug discovery. You know, there are a lot of problems, why drugs are expensive, why they come really rare. So I want, because I'm a retired guy, I can speak about my own mistakes of big companies, small companies, as well as university professors. We all make mistakes, but I'm hoping that we are learning from the mistakes, or you will learn from the mistakes. This is the purpose, or this is the message I want to give you. Not that you learn everything about the drug discovery. So, as I promised Şevket, here is my 4 universities academic career, if you like. But the reason of this slide is this. The 30 years I spent, I started with Novartis 18 years in Basel, and then there I became a drug discoverer. I worked on several different departments, and then moved to New Jersey, Avantis 5 years, 3M 3 years, and Alba Therapeutics, which is, this was 70,000 people, this was 120,000 people, but this one had only 40 people. So I wanted to know how, whether small is beautiful, okay? So with that experience, I hope to give you some idea about drug discovery. But before going into that, I think we need to know a little bit about the medicine in general. Then I want to go into really details of classical drug discovery, as well as problems, and I have some suggestions. Okay? If you have a hardcore pharmacologist among yourself, I hope we will have a nice discussion. If not, don't believe everything what I say. Just say there must be something wrong, otherwise I won't enjoy our discussions at the end. Then I will go finally to new paradigms, my suggestions, how can we become innovative and better drug discovery. Okay? So, expectation from society, from us, scientists, is, tell me if you have something to add. They expect that we cure cancer because the number of the cancer patients increasing like infectious disease. Find better anti-infectives because infectious agents are mutating and they are becoming resistant. Better drugs for allergy autoimmunity. Funnily, allergy and autoimmunity increases. People think because of our genes, I don't believe. I just gave a talk 2 days ago about this big allergy asthma meeting, saying no, our genes did not change in 30 years. Something else has changed. That's our environment, lifestyle, whatever. Find magic bullets for curing, that is good drug delivery, so that it meets the organ rather than disseminating everywhere, affecting your brain when it should be in the liver. And major stuff, again, you have to get non-invasive for the body. And of course, finally, if you can, you should be repairing genetic defects. How many genetic diseases exist? My wife is a geneticist. 4,000, 3,000, 3,000? How many?
It is more than 3.
More than 3,000, but at least 4 or 5. People already know exactly what the defect is, and they are transferring genes, and these genes are repairing, but there are problems. They cause problems. It's not easy. So this is the general expectation. So how many different ways of drug discovery exist? And here, very simplified, I think we should look at the folk medicine first, right? They folk knows and they use it, and again, it has something to do with Basel. Somebody in German university actually discovered that this bark tree, I believe, has aspirin in it, and finally it's synthesized acetic acid. And then there are many, many drugs discovered serendipitously. That's where Novartis, I give you this example, because this was discovered or synthesized for a total different reason. A university guy at the Australia discovered that it chelates iron. So hemophiliacs, they have a lot of iron and they have a lot of problems. And the drug removes iron. It became, say, multi-million-dollar drug, if you like, by chance. And there are, the cyclosporine, organ transplantation, rejection of heart, transplanted tissues, you need to suppress the immune system of these people so that they don't reject the organ. Cyclosporine is discovered. This is a natural, huge, 10 amino acid cyclic complex drug present in nature, fungi, I believe. And there's a beautiful book, a very nice story how it's discovered. A lot of fight, human nature. And this was discovered by secreting the whole animal. Somebody injected the drug into the whole animal and tried to find out whether it suppresses the immune system or not. And this is now multi-billion-dollar drug. Over 40 years, there's only one coming close to this. Not a single, another drug has been discovered. It is rare to discover such a huge, success drug, by the way. And people become sophisticated. They have their cell targets. They screen thousands and hundreds of compounds using the cells of interest. And of course, the best is molecular screen, and the Gleevec, is a big success of Novartis again. And this is a tyrosine kinase inhibitor. And by chance, somebody discovered at the university. They were looking for other things. It was designed for cancer therapy, not for this cancer, CMA, chronic myeloid leukemia. Okay? There are some serendipity here, but definitely there was a thinking behind. We knew the enzyme structure, and computational chemistry has been used for years. Okay. This is just to remind you that we should be looking at the Mother Nature. Because 2 reasons. First, Mother Nature makes complex drugs. Chemists make simple ones. They make millions, but they are simple. And simple drugs, of course, are good for kinases, but many other things cannot be inhibited by simple chemicals. Therefore, we should not forget the Mother Nature and look at it. And big companies already realize they are buying huge forests. A future Turkish company may buy the surrounding forest here just to collect the leaves and the roots, whatever. We will screen. I don't know whether this is happening, but entrepreneur, we will come to that in the end. We'll do that one day. There will be a lot of interesting stuff. Grouping of drugs according to their targets, as you know, these are enzymes. For example, COX inhibitor, aspirin is COX 1 and 2 inhibitor. Voltaren is COX, whatever. There are so many different COX inhibitors. And substrate inhibitors, receptors, probably 60% of the drugs are GPCR inhibitors. This is a very easy, nice target, apparently. Ion channel inhibitors, huge market, huge success. And RNA, DNAs, and various physiochemical mechanisms. But also lately, big industry is on the biologicals, cytokines, monoclonal antibodies, and others. Okay? I think this is enough as an introduction, just to think you about the drugs and what the society expects. Now, let's go to more difficult things. How do we discover at the moment? Oh, let's go to that discovery. How is it done? What are the problems? And I would like to make some suggestions. And a cartoonist made this beautifully. This is such a complex discovery process that you can fish yourself, your leg, instead of or your toe, instead of a drug. Believe me, this artist is very clever. This is what is happening, you know. When you know everything, you will understand why he made it. But sincerely, look at this complexity nowadays that we have. We have genomics, proteomics, biochemistry, immunology, biochemistry, and many scientists come together, form a team. Team says, for example, you want to treat what? Rheumatoid arthritis, allergy, asthma. Just you choose. Then you need a disease hypothesis. Why do we get asthma? I just give you an example, but you can pick up another one. So you need a solid scientific background about that disease. This is where the problem starts. Usually this disease hypothesis, knowledge about the pathogenesis of a disease changes as new information becomes available, right? But I have to have a drug today. The patient is unable to breathe. They have a problem in breathing, right? Then you can say, yes, I have a drug, beta agonist, corticosteroids, relax, they cannot breathe. But the disease continues for lifelong because you don't know underlying pathogenesis. So I suggest 10 other targets, which is correct. We'll come to that, how do we do the selection. But this is how you select, you can say method of protease inhibitor, I don't know. Or there are 10 different cells. You can inhibit the eosinophils. I didn't do it, but a company called, I don't want to give you a name, spent millions inhibiting eosinophils by making antibody against IL-5, which is the growth factor for eosinophil, because the most famous, the most powerful, at least 5 professors in the world said, everybody, this is the cell causing asthma. And the company spent all this money, made a lot of experience, went into sheep models, mouse models, even monkey models using ascaris. How close you can come to human disease?